in fact, the technique, all the techniques I'm going to mention in lab tomorrow, but that sort of lead us into the big one, are related to antibodies. And today we are going to talk about antigens and antibodies. So thus far, we have been talking about the innate immune response. We've been thinking, we saw first sort of that very immediate part of the innate immune response that happens within the first few hours following microbe coming into the body, where we are using those preformed proteins. We also talked about the cells of the innate immune response that start acting a little bit later to lead to their responses, and that's because those cells needed to get activated and do some signal transduction, so it needs a little bit of time, unlike the preformed proteins. And in both cases, sometimes the pathogen can be eliminated with the response, and we live happily ever after. But in other times, we are not going to eliminate the pathogen just with those innate immune responses, and we're going to need to have the adaptive immune response working as well. And so in starting to talk about antigens and antibodies, we are starting into thinking about the adaptive immune response. In telling you about this, I want to tell you about some experiments that were done by two scientists in the 1890s, von Behring and Kitasato. Von Behring and Kitasato won the Nobel Prize for these experiments. These are one of many Nobel Prize experiments that we're going to hear about this semester. And I'm going to describe these experiments like they were all in one paper. If you tried to find the paper, you actually would find out that I'm combining stuff from a few different papers here for ease of our discussion. So I apologize if you desperately want to read it and then can't really find the details to read it, because it's not one paper that I'm throwing some stuff together. These experiments were important in some of our understanding of particularly the parts of adaptive immunity that we are talking about right now. I will admit that they leave some stuff out. When I sort of go through the specifics, I will mention, it will be less important to you, exactly which pathogens von Behring and Kitasato used in their experiments. And we now know that they lucked out in terms of they just picked some pathogens that seemed cool to them. The pathogens they picked all have some unique features that made this experiment really clean and really only involve one part of the adaptive immune response. So yes, this does kind of oversimplify some things because of the pathogens they picked, but it's useful for our purposes right now. So von Behring and Kitasato did experiments using 3 different microorganisms. The 3 different microorganisms are Corynebacterium, diphtheria, Clostridium tetani, and Clostridium botulinum. Again, we now know that they kind of lucked out in that there's something that these have in common. Corynebacterium diphtheriae causes a disease called diphtheria. Clostridium tetani causes tetanus. Clostridium botulinum causes botulism. I'm only going to mention Clostridium tetani and Clostridium botulinum. I'm not going to go through the experiment with diphtheria as well. And the reason why I choose Clostridium tetani and Clostridium botulinum as my examples that I'm going to go through here, is because you can notice I actually have the scientific names for the organisms written here. They're not in italics because I don't know how to write in italics. But what you can notice is that these two organisms are in the same genus. So they're pretty closely related, though they are different organisms that cause different diseases. I'm pretty much just going to call them tetani and botulinum, to make our lives sort of easier. And the first thing that they did is they took a rabbit, and they took a syringe and infected the rabbit with a low dose of Clostridium tetani. Yes, I once had a student ask me why I drew a picture of a baseball cap getting hit by a shovel. But it's not a baseball cap getting hit by a shovel. It's a rabbit. And it's a syringe. So the rabbit was infected with low-dose Clostridium tetani. And after that, the rabbit lived. Here, I'll give it a carrot. They then did a different experiment where they had another rabbit that was given a high dose of Clostridium tetani. And when they did that experiment, the rabbit died. That's my dead rabbit. It can be sad. So they did this experiment. They actually did the identical experiment with a low dose of Clostridium botulinum, and the rabbit lived. They did a high dose of Clostridium botulinum, the rabbit died. They also did the same low dose, high dose with the other microbe. Low dose, rabbit lived. High dose, rabbit died. And then they were running out of rabbits. So they did another experiment. They took this rabbit who had lived through low dose Clostridium tetani infection, and they gave this rabbit a second infection. And now they gave this rabbit a high dose of Clostridium tetani. You can remember that in previous experiments, if a rabbit got high dose Clostridium tetani, that the rabbit died. However, in this experiment, they saw that if the rabbit had previously had a low dose of Clostridium tetani, before it got the high dose of Clostridium tetani, it was protected. Somehow, previously having lived through a low dose protected the rabbit from a high dose of Clostridium tetani. And so that was cool. But they also did one additional experiment. Actually, they did two additional experiments that we're going to think about. So they also took a rabbit that had gotten a low dose of Clostridium tetani, and they gave that rabbit a high dose of Clostridium botulinum. So high dose, again, this is the dose that kills the rabbits. And this is a member of genus Clostridium, but it's not the same microbe that this rabbit had previously seen. So you can now see rabbits getting high dose of a different microbe. And in this case, the rabbit died. And so what they learned here is that having previously had a low dose of Clostridium tetani didn't like magically protect you from everything. It only protected you from the same thing over again. If you were challenged with something different, even something pretty closely related, a bacteria in the same genus, it was not protective. And so, one of the things that they took home from this experiment, and one of the things that we talk about with this experiment, is the idea of specificity in the adaptive immune response. And so they saw that this magical protection was specific to a microbe that you had seen before, and wasn't just like your immune response improved generally. And so you can see here are molecules that are binding to one virus, measles, will not be able to bind to a different virus, influenza. And both of these are RNA viruses, so they would actually both trigger the same PRRs. And so one of the things that was learned from the von Behring and Kitasato experiment is this immune principle of specificity. For reasons that I've never understood, immunologists really love to use the phrase, the exquisite specificity of the adaptive immune system, because it's so specific. This is the this variant of COVID versus that variant of COVID kind of distinction that these responses are making. They're really, really, really fine distinctions. But then von Behring and Kitasato, and in fact, did one more experiment, and other scientists extended those experiments from here. And because what they saw in this experiment was sort of like that this rabbit had like magical protection. Once the rabbit got this high dose of tetani, after previously having low dose, the previous low dose had like gave it magic protection. And so they wanted to try to figure out what was giving the rabbits protection. At this time, immunologists were kind of arguing a lot about whether the immune system was proteins in the blood, or was magic stuff in the blood. Actually, they didn't know it was proteins. Whether it was magic stuff in the blood or whether it was cells. Now we kind of understand that distinction slightly differently, but that was what they were like trying to think about at that time. And so von Behring and Kitasato wanted to say, okay, so we have this rabbit getting this magical protection. Is the protection from blood stuff, or is the protection from cells? And so in their experiment, they did this one final experiment. So they have the rabbit. Rabbit gets low dose Clostridium tetani. And then they euthanize the rabbit and take samples from the rabbit. And they take two kinds of samples. One kind of sample is a serum sample. And so on this slide you see some pictures of serum. And basically serum is this liquid part of your blood. So if you take blood, let it clot, let all the cells come out and just have the liquids and the proteins and also the lipids actually. All of that stuff at the top. So it's like the part of the blood that's not cells. It's the liquid and everything else is the serum. And you can basically get that, you can do a separation of blood and get all the cells in a pellet at the bottom and all the liquid. And that liquid is called serum. So they took serum, which to them represented, the liquid blood stuff. Or they took the spleen from the rabbit, very similar to what you guys did in terms of isolating a spleen and making a cell suspension of a spleen, like you did in lab last week. And they took those two samples and they put them in other rabbits. These samples were from a rabbit who had received the low dose. So the idea is now the second group of rabbits hasn't actually ever been infected, but has sort of gotten either the liquid or the cells from a previously infected animal. And so what they're hoping to figure out is which one had the magic protection. Was it the liquid part of the blood? Was it the cells of the blood that gave this magic protection against Clostridium tetani? If I did this experiment in my lab right now, I would never have picked Clostridium tetani because I don't really have any particular affinity for Clostridium tetani, and the experiment would have been a mess because I would pick a microbe that has different properties. They happened to pick a microbe that had such good properties that they got a very, very, very clean answer. So for some microbes, the answer about cells versus liquid, the answer would be both. But that was not the answer they got because we now know, lucked out in terms of which microbes they picked. And what they saw in this experiment was that the rabbit who had received the serum lived, so it gets a carrot, and the rabbit who had received the spleen cells died when challenged with high dose tetani. This type of experiment where you take some immune cells, potentially from an organism with some experience, immune experience, and transfer it into another animal is a thing that immunologists do all the time. We do stuff like this all the time. It's officially called an adoptive transfer. And this is one of many adoptive transfers that you might see. But the conclusion that they came up with from this experiment was that in the case of Clostridium tetani, also botulinum and diphtheriae, the magical protective thing was something in the blood. It wasn't a cell. It was something in the blood. We now know, spoiler alert, that the magic thing in the blood is an antibody. So antibodies are in fact the answer. I'll get us more to some experiments about how subsequent scientists found that the protective thing was antibodies. And some details about antibodies in a little bit. But before I do that, I would have one other sort of thing I want to tell you about. Because you might look at this experiment and you might say, okay, yeah, great. 1890s. This does not matter at all today. She's telling us about old, dusty experiments no one cares about. Early Nobel Prize, blah, blah, blah. We still do something based on this experiment in medical fields today. So we actually do something called passive antibody therapy today. We know now that the magic stuff is antibodies, and that sometimes individuals or animals will have a lot of antibodies in their blood. And we know that we can actually transfer those antibodies into somebody and help them out. And so if, for example, you get a black widow spider bite, what they will do is they will give you a shot of antibodies from someone else. So those antibodies can immediately protect you. You don't have to worry about a cell making anything. You basically are getting some antibodies from somebody else. And so the idea is there's going to be some antibodies that were taken from someone else, that are going to be injected into you to protect you immediately. It's passive because you didn't make those antibodies, somebody else did. So it's this passive antibody therapy. And you can see a number of situations where passive antibody therapy is still used today. So you can see, black bite or spider bite. You can see, snake bite. If you actually get tetanus, this is what will happen. If you've ever heard about, what happens if you actually find out that you got infected with rabies and you have to get the shots afterwards, which are like really not fun. That's because they're giving you passive antibody. And so this idea of passive antibody therapy, of transferring antibodies from a previously immune individual, is still used all the time. If you actually think back to the very beginning of the COVID pandemic, the first treatment that was used effectively in COVID pandemic was blood from people who lived through COVID. So you might have heard about, there were some things in like early 2020 where if you'd had COVID and lived through it, they asked you to give blood. And the reason was because they were going to do passive antibody therapy and give your antibodies to people who were in the hospital who were really sick. This is also one of the big things that was done that's done in big Ebola epidemics. And so we still do this, and it's really all the same thing that you saw with Von Behring and Kitasato's experiments. In fact, you might have heard of some of the treatments. This is particularly before Paxlovid for COVID. Some of the earliest treatments were actually that we'd figured out how to make the antibodies in the lab instead of having to get them from a person. And so the first treatment that we actually had for COVID was that we just made the antibodies in the lab and we're doing passive antibody therapy. So if you've heard of, for example, bamlanivimab, that was just an antibody we made in the lab to do passive antibody therapy for COVID patients. And there are some other companies made antibodies as well. So, while, yes, I'm telling you about a set of experiments that were done in the 1890s, and you might be like, no one cares. In fact, we still use, exactly the same principle from those experiments today. So people after Von Behring and Kitasato looked at this experiment and thought it was cool. And they said, all right, we know there's magic protection. We know it's some part of the blood that's magic protection. But we want to narrow it down and find out what part of the blood is actually giving us this protection. So the first thing that people did was they treated the blood with different enzymes. So one enzyme that they treated, or specifically they treated the serum with different enzymes. One enzyme that they used was a lipase. What does a lipase enzyme act on? Yeah? It breaks lipids. It cleaves lipids, yeah. So if you use the lipase on the serum, you basically destroy all the lipids. So they did that, and then they did the same experiment, and they looked to see, does the serum still work? And that was basically them trying to say, is the magic thing a lipid? Or did our lipase destroy it? When they did that, they found out it wasn't a lipid. And they did the same experiment with a glycosylase to destroy carbohydrates, and they found out it wasn't a carbohydrate. And they did the same experiment with nuclease, and they found out it wasn't a nucleic acid. And they did the experiment with protease, and they found out, oh my gosh, the magic thing is a protein. There's some protein in the serum that is actually providing this protection. And again, it was really the same experiment over and over and over again. That you see here. It's just breaking up the serum more and more ways to try to figure out what is the magic protective thing. So then they knew it was a protein. Yay. And then they wanted to know more about this protein. What is this protein? And at that time, they weren't like amazing, awesome biochemists. But they knew they could divide the blood into 4 proteins. They knew there were 4 major protein groups in the blood. One of them was called albumin, and it was like a massive protein in the blood. Just so you know, this data from this textbook, this is actually how they separated it. These 4 peaks are the 4 different proteins. This is what they did before a gel. So these would have been 4 bands in the gel, but it was before they did gels. So they're like, well, there's this one protein called albumin. There's a lot of it in the blood. We now know there's a lot of albumin in the blood. And we also got these other 4 proteins. And they're globular. That's basically just a biochemistry term. Don't worry about it. They're globular, so we're going to call them globulins. And there's 3 of them, and we don't really know anything about them, so we're going to call them alpha, beta, and gamma globulin. So globulin, and like I said, it was either alpha, beta, or gamma. And they did experiments both where they transferred each of these fractions, just like they had done in the previous experiments, and they did some experiments where they tried to make the magical protective thing go away, and looked to see which one was missing to try to figure out what was up. And in fact, when they did that experiment where they got rid of the magical stuff, they got the results shown in black. And the result shown in blue is normal. And so which one seems to include the magical protective stuff, if black is where it was gone? Yeah, David? So it looked like gamma globulin was the one that was the important one. In some ways, they also said, aha, so we found out that gamma globulin is the immune globulin. It's the one that has the immune powers. If you go to the hospital today and you need antibody treatments, you need antibodies put into your body, you know what they're going to say what kind of treatment you're getting? You're going to go to the hospital and get gamma globulin treatment. It's still what it's called today. Like, that is a very common treatment that some patients will get is gamma globulins. Sometimes people will also talk about immune globulin because it was the immune one of all the choices. It was the one with the immune activity. And the term that immunologists tend to use very frequently when they are talking about antibodies is immunoglobulin. So we get this term that we're going to use a lot for antibodies of immunoglobulin just by smushing together those 2 terms. I totally spelled it wrong there. I don't know what that is. Globulin. I definitely know some people who are immunology trainees and students who call themselves immunogoblins, based on this. So after all of this stuff, people actually were able to start looking at these proteins, these immune globulins, under electron microscopes. And you can actually see 2 of those early electron microscope pictures here. So those are actually some of the earliest electron microscope pictures of antibodies. And what you can see is it looks like a Y-shaped protein. And so here is our antibody, and what that protein looks like. You can see that antibody structure here from your textbook, and I'm going to talk a bunch about details of this structure and what we can see with this structure. But one of the first things that they found when they looked under the electron microscope at an antibody was that this Y-shaped structure had basically two arms that were identical that could bind to things. And so they said, okay, we've got these two kind of binding arms and they're identical. So there are actually some images where people like have stuff bound at the end, and they could find, you always get binding on these two arms, kind of like these two so much shorter arms, and you don't get binding to your thing and this tail, and the same thing binds to either one of these arms. It's not like it's two separate binding sites that bind to unique things. It's duplicates. And this thing that the antibody binds is known as an antigen. And so I need to tell you a few things about antigens. And specifically here, I am telling you about the antigens that are bound by antibodies. When we get to T cells, the antigen there will be some differences in the antigens. In fact, sometimes I like to ask questions about like comparing and contrasting the antigen with antibodies and T cells. I will also tell you that the way that, if you look at the syllabus, the way that things are structured is that I've sort of introduced the idea of an antibody. I'm going to tell you some stuff about the antigen that binds to antibodies, and then we're going to swing back to more about the antibody part after we know some stuff about antigens. We're actually going to do that same sort of structure of timing when we do T cells in terms of the T cell receptor and then the antigen. It's just that it will feel different because that antigen is more complicated. So we need to think now about our antigen. And the antigen is a molecule that is recognized by the adaptive immune response. And so usually if we're talking about the innate immune response, we talk about a PRR and a MAMP. So the MAMP is the thing, the receptor is the PRR. Here, if we're talking about the adaptive immunity, we talk about an antigen instead of a MAMP. And one of the really important things to know about antigens is something that we know based on this experiment that was done by a scientist named Carl Landsteiner. Landsteiner did not win his Nobel Prize for this experiment. He won it for a different one. He also is the person who came up with the ABO blood groups and discovered all of that. That's where he got his Nobel Prize from. But Landsteiner did this experiment that was pretty important in helping us understand antigens. Particularly, we now know antigens that bind to antibodies. Landsteiner did a bunch of organic chemistry. And he made these 3 dyes that you can see at the top here. And what I want you to imagine about the 3 dyes, or the 4 dyes, sorry, is that they had never been made before in the history of the world. So I want you to imagine Landsteiner, this is like the first time they're ever made. Was for Landsteiner's experiment. And Landsteiner actually made them and attached them to a protein, but we're going to not deal with that right now. And then he took his 4 dyes and injected them into 4 different groups of animals. I don't remember which animals he used. But he injected them into 4 different animals. And he was able to find out that he could get an antibody response to these dyes. So these, we'll call them rabbits. I don't remember what they were. These rabbits made an antibody response to dyes that had never before been synthesized in the history of the world, which gave us one of the first key things that we learned about antigens that bind antibodies. And that is they can be literally anything. Any sort of chemical molecule that you can imagine, whether it's been synthesized or not, you can make an antibody to. And in fact, you can see these rabbits, they made these antibody responses even though these were brand new dyes. So like, I made a dye tomorrow, you already can have an antibody response against it. That's one thing that's like so cool about these responses, is that we get antibody responses to literally anything. But he also had one other key observation from this experiment. So in this case, I want you to imagine we've got these little dyes and they're added to a great big, there's like some giant protein attached to them. These rabbits, we're calling them, are making an antibody response. And when he looked at the antibodies that he got from the rabbits injected with this dye, he found that those antibodies bound to this dye and did not bind to these dyes. And what you can notice is that these dyes are actually quite chemically similar, especially if you think about this also like this whole giant protein attached. There's only this one carboxyl group difference. And when he made a version with the carboxyl group, the antibodies only recognized this version and not these with the carboxyl group in slightly different locations. And so he also learned just how specific antibodies are, because he was able to see that even these really small chemical differences could be distinguished by the antibodies. And when you make an antibody to one of these antigens, it's actually unique to that antigen and not reacting to these very similar antigens that are shown here. So one of the things that we learn from this is that if the antibody is binding to this protein with the dye attached, it's probably not like going and hugging the whole thing. It's got to be able to bind where this dye part is and actually do some distinguishing. And so we often think about another term in relationship to antigens, which is something known as the epitope. And so often I, as an immunologist, think a lot about epitopes. Epitope is the part of the antigen that is actually making physical contact with the antibody. When we get to T cells, it will be this actual portion of the antigen that's making contact with the T cell, but same basic idea. And so this whole molecule, this whole yellow molecule, we will say it's a protein. But in the case of antibodies, it could be a protein, it could be a lipid, it could be a nucleic acid, it could be a carbohydrate, it could be a random dye. This whole thing, like I said, I'm going to call it a protein, but it could be any of those things when we're talking about antibodies, is the antigen. But there are different sections of it that might bind to different antibodies. And so here you can see there's this little patch that's made up of these 3 loops, and that little patch binds to the blue antibody. It's just perfectly complementary to that blue antibody, and so we refer to this as the epitope for this antibody. So there's sort of this blue epitope. Here you can see, here's a different patch that is binding to the red antibody, and so that would be a different epitope. And so you can see that each antibody will bind to a particular epitope, and you can also see that any one antigen may have multiple epitopes. So in the case of my antibody here, the epitope is this sort of roundy side. The epitope isn't the eyes. That's not where it binds. It binds here. So this section is the epitope. The whole thing is the antigen. One thing I've already said to you on the previous slide that I will say again because can't say it enough is that when we are talking about antibodies, the antigen can be anything. The antigen can be a protein. The antigen can be a carbohydrate. The antigen can be a lipid. The antigen can be a nucleic acid. It can be literally anything, as Landsteiner showed us. Please note, when we get to T cells, that's another big compare and contrast thing, is all of those things can't be antigens for T cells. So that's why I'm hitting the fact that all of them can be for antibodies really hard. This example also shows you examples of protein antigens. And what I want you to notice about my protein antigens that are binding to my antibodies is that the proteins that I'm showing you are not sort of linear primary structure proteins. They're not like a straight line of amino acids. They're folded in some way. And antibodies, we tend to think about as binding to proteins or whatevers that are sort of in their three-dimensional structure, their three-dimensional context. So that means if you were to unfold these proteins, if you were to denature them, you might actually kill the epitope. You might kill the ability to bind. And so the top here says antibodies recognize native epitopes. That means antibodies recognize epitopes in things that are folded correctly. Now you can notice the one on the right. If I unfolded this protein, these two amino acids wouldn't be so very close next to each other if I just pulled the string and made it straight. And the antibody probably couldn't bind anymore because it wouldn't be in the right geometry. So I actually would have ruined this one. Sometimes they might bind to some amino acids that are next to each other. And so this one, if I unfolded it, it'd probably still work. This one wouldn't. So whether or not unfolding totally kills everything kind of depends on your epitope. Specifically, it depends on, is that epitope an epitope that is just a line of amino acids in your protein? If it's a protein antigen, that would be called a linear epitope. Or is your epitope an epitope that only is found when you have a folded protein or folded whatever antigen? That might be a conformational epitope. So now when I unfold this protein, you can see the loops aren't next to each other anymore, so the antibody won't work. If you see this and you say, you know what, I'm never doing immunology ever again. But you do spend some time doing any kind of biomedical science or any kind of biochemistry, this will be the most important slide for you to know. Because if you do any technique that uses an antibody, like a Western blot, you need to know if your epitope is a linear or conformational epitope so you know how to prepare your samples. Because if you do something like unfold your sample and then use an antibody that only recognizes folded sample, you're going to get a negative answer no matter what, and it's only because of how you prepped your sample. So this actually is something that's super important for throughout all of biomedical science. Whenever I think about an individual antigen, this has been implied in some of the previous slides, but I will say it explicitly now. Any antigen can contain multiple epitopes. So each antigen doesn't necessarily have one epitope, it has many. That might be repetitive copies of the same epitope, like you see on the right. or that might be multiple different unique epitopes like you see on the left. So I'll say this on the later slide. In addition, if I actually go not to an antigen, but if I go to the level of a pathogen, like a whole microbe has many antigens. And then those antigens all have multiple epitopes. So individual pathogens can have multiple antigens, and individual antigens can have multiple epitopes. So to give you an idea of what this looks like, I've got two examples. On the left, you can see a cartoon version of HIV. And you can see all sorts of different parts of HIV that are color-coded and labeled here. Lots of different proteins, nucleic acids, lipids, all sorts of good stuff. Each one of those could be an antigen. So GP120 is an antigen. GP41 is an antigen. P17 GAG is an antigen. All of those are antigens. You can see this one microbe has so many antigens. And if this is actually a zoom in of GP120, just this yellow protein that's shown here, all of these little patches that are shown in different colors are different epitopes that we know of in that one antigen. And so I could give you a list of sort of all of these patches for every epitope in every antigen. So what I want you to realize from this is that you really aren't making one kind of antibody responding to HIV. You're actually making tons of them. You're making one antibody to each epitope of each antigen. So you've got a lot of sort of target. Those are the targets. You have a lot of bow and arrows to hit the targets. I don't know what my term is. But you have all of these different epitopes within a microbe. I mention this because if you think about what's happening in your body when you get infected with a particular virus, I'm going to frequently mention, and then you have the antibody against influenza. Not right. There's not one antibody against influenza in your body. There's one antibody against every epitope of every antigen. So there might actually be like 1,000 unique targets in influenza, and you actually have an antibody against every single one of them. That's just a lot of words to say other than the antibody against influenza. And so what I want you to see with these epitopes is what I mean by specificity. That you have that each antibody has specificity for one little epitope in each microbe. Similarly, on the left here you see SARS-CoV-2, the virus that causes COVID. You can see all sorts of different parts that are color-coded here. And here you can actually see on the left one antigen from that virus. That one antigen is this yellow thing called spike. This is kind of also interesting because when you get the vaccine, this vaccine, we'll say so much more about vaccines later, but when you get the SARS-CoV-2 vaccine, you're actually only getting the spike part. You're not getting any of the rest of this. Yay, safety. So you're only getting spike. You only get one antigen, not all the antigens. But it turns out even spike, we have been able to find many epitopes in it. So you're not just going to make one antibody in response to that antigen. You're going to make an antibody in response to every epitope within spike. If you get infected with this virus, then you're going to make an antibody against every epitope in the whole virus. And in the past couple of years, we have actually been able to do things like start to count how many epitopes are there. This has, only been a recent thing that's been going on during the pandemic. You can see this is a paper from 2021. But just to give you an example, we're pretty sure that within spike alone, there are 20 antibody epitopes that we can then add on to some other types of epitopes. And so when you think about your antibody to the vaccine or your antibody to spike, really you actually have 20 different ones that are each binding to different targets on spike. Yeah.
Why is it not? Why is it
not? It's a great question. A lot of it has to do with sort of structural biochemistry stuff in terms of both where the antibody can access and some of the details of how antibodies are generated. And so it ends up not being infinite because of some of the details of antibody generation that we'll talk about later, but also some of the details of sort of biochemistry structure of what you can or cannot pull off. But imagine it's a big number. Yeah, Jonah.
Is there like an antibody self-recognized self thing that it doesn't always sort of detect your cells versus?
So Jonah is very worried about antibodies detecting self, and it's probably because you have many biomolecules, proteins, lipids, carbohydrates, and I told you like how broad recognition is. The answer is yes. So this is going to be a 2 weeks from now problem, I think. But what I'll tell you now, and again we'll see much more of later, is that you actually make those antibodies and then kill the cells that made them. So you actually do a testing process to kill any cell that makes a self-reactive antibody during the antibody development process. And so that also gets to some of your question, Brian, is that there may have been more epitopes, but we may kill those cells. And so that may be part of it because some of those may be self-reactive. Yep, David.
Because following that, like when it comes to like the dyes, like example, do we have like antibody responses to certain medicines?
Yes, absolutely.
Does that ever impact like their?
Yes, so there are people who are medicine allergies. That's largely going to be an antibody response. Again, this is going to be sort of more of an allergy thing, but basically what you saw with that dye thing is kind of similar to what happens. I'm like months ahead of myself with this sentence. With poison ivy, is actually you have a compound from poison ivy that's modifying another compound. I have an allergy among my many allergies to nickel. So if I wear cheap jewelry, I get a response. It's the same. It's the same kind of thing. So yeah, you can make antibody responses to all of those types of things. Yes. It's okay.
But then for the, when we're talking about the COVID vaccine, like when you have like the COVID boosters, did that like contain like the same thing?
Great question, and that gets a point I was like deciding whether or not to make. So perfect. Sometimes people have been like freaking out about different COVID variants, right? Or they'll be like, oh my gosh, this COVID variant avoids antibodies. Actually not true. What actually happened, and I read the original papers, is that they will show that a particular variant has changed one epitope and can avoid antibodies at that one epitope. But the other 19 are still fine. And so yeah, you're avoiding one of the 20 antibodies with that particular variant. But you still got the other targets that are not moved. And so yes, it does avoid immune responses. But if you actually think about it in big picture, it's not really avoiding all that much. And so that led to some debates about with boosters, do we need to give you the new versions of the virus or not? I know people who still debate both sides of that. But the answer is we are giving you the new versions. So all of this is awesome stuff. So one other thing I just want to tell you, on this slide and sort of the next couple slides, this is all sort of related to some stuff we're talking about, is the antibodies that we are talking about, these lovely proteins, are made by a particular type of cell. And that cell is called a B cell. And so while we are talking about antibodies a bunch right now, technically we're kind of also talking about B cells, because B cells are the ones that make these antibodies. And as I am talking about antibodies for the next section of the course, there are certainly situations where I'm going to just slip and talk about the B cell. And I want you to know why that is. And in fact, there's one other part of the reason why that is. B cells have a unique receptor on their surface. It is very creatively called the B cell receptor, or the BCR. The B cell receptor that a particular B cell makes is just an antibody with a transmembrane domain attached. So sometimes when I think about making antibodies, I'm also really, well, not sometimes, all the time when I think about making antibodies, I'm actually also thinking about how we make B cell receptors. And sometimes when I say something, I might mess up at some point in describing stuff and say the B cell receptor, blah, 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 when I should have said the antibody, blah, 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 blah. And the reason why I might mess that up is because they're actually the same thing. This also gets me to one other kind of thing I want you to know about B cells. It's a big principle of adaptive immunity. And it's, again, one of those things that I can't say enough. I'm going to I officially introduce this concept to you later, and you're going to find it terribly confusing. So I hope that it's not confusing now, and you can be like, oh wait, it's the same thing. I get it now. Each B cell makes one antibody / B cell receptor that can respond to one epitope. So each B cell is unique. So I mentioned that with SARS-CoV-2 spike, you've got 20 antibody epitopes. If you were going to have an antibody response against SARS-CoV-2 spike, you would need 20 B cells participating, each of them having its own unique epitope that it recognizes. So each B cell is really only recognizing one epitope. You can see this here as well. We will have one B cell might have multiple B cell receptors, but they're all recognizing the identical epitope. In fact, even the two arms of this antibody are both recognizing the identical epitope. So we have this homogeneous binding. The difference here is that each B cell will make a slightly different receptor. So you've got each B cell has its own unique receptor that just belongs to that B cell. And eventually that B cell may divide a bunch of times and make more copies of itself. Sort of like Attack of the Clones. It makes a bunch of clones of itself. And they will all have that same specificity. And so this process where we have a whole bunch of cells that have one specificity and only one specificity, and then those cells might expand out, might make a clone of themselves, is sort of the underlying way that all of adaptive immunity works. This is officially known as the clonal expansion theory, clonal expansion. And I want you to compare and contrast that to what we saw with our macrophages and their PRRs. Remember, every macrophage had tons of PRRs. So every macrophage could recognize any microbe that happened to come around, because that macrophage would have all sorts of different PRRs. But each B cell is like specific to this variant's COVID, this part of spike. And it's going to be useless against Ebola. while the macrophage has all sorts of PRRs and is going to respond to whatever. And so this is really where that specificity comes from in the immune system. One other really important piece one other really important thing that is related to all of this is, again, a lab-ish thing. In that we often will think about two different kinds of antibodies. If you are going to go do a Western blot and you need to buy the antibodies you need for your Western blot, one of the things you will have to decide is if you want to buy a polyclonal antibody or a monoclonal antibody. So I have to tell you what polyclonal versus monoclonal antibodies are. And so I want you to look first at this list of polyclonal antibodies. Polyclonal antibodies are like real life antibodies. The idea of a polyclonal antibody or a set of polyclonal antibodies is that they are antibodies that all bind the same antigen, but they might bind different epitopes of that antigen. So another way you can think about that is that polyclonal antibodies are a mixture of many different antibodies that came from many different B cells. They have in common that they all bind to the same antigen, but they might bind to different epitopes. And so here you can see that my mixture of antibodies, my polyclonal antibodies, includes the red antibody. It came from the red B cell, not pictured. It binds to the red triangle antigen. You can see that my mixture of polyclonal antibodies includes the blue antibody, which came from the blue B cell, not pictured, that binds to the blue epitope. And you can see that it also includes the black antibody, came from the black B cell that's not pictured, and binds to this gray-black epitope. So this is sort of the idea of what polyclonal antibodies look like. And what you can imagine is if I inject any antigen into you, if I give you any vaccine, that's actually what happens. That's what you make. You always make polyclonal antibodies. We have figured out a way in the lab to make antibodies where we don't get a mixture of antibodies that all bind to different epitopes within the same antigen, but where we get a purified population of antibodies that all bind to exactly the same epitope. These are known as monoclonal antibodies. And to do this, we actually had to find the blue B cell and isolate the blue B cell and get just the antibodies that were made by the blue B cell, instead of seeing the whole population that we really see in an organism. And there are lots of situations where in experimental settings, we like to use monoclonal antibodies. When we do some of those therapies that I told you about, we use monoclonal antibodies. Like in a lot of our patients, we're using monoclonals right now. And so in a lot of situations where we use these in people, we use monoclonals that we've made in the lab, where we're getting a purified population that's responding to just one epitope of the antigen, instead of kind of the real life scenario of what you get in a polyclonal response in your body. I will see you guys on tomorrow. That's a weird grammar. I will see you guys tomorrow when we're going to talk about a particularly important technique that involves antibodies called flow cytometry. And and then we will continue talking about fun antibody biochemistry on Friday.